Good morning, everyone. I am from biology classes. Today I am going to discuss about the another topic of photosynthesis, that is chemiosmotic hypothesis or chemiosmosis. Okay, how the ATP can synthesized inside the chloroplast. It was first studied by Peter Mitchell. How the ATP can generated inside the chloroplast, which are utilized for the synthesis of glucose as energy source. This ATP generation inside the chloroplast was first studied by Peter Mitchell, and he put a theory or hypothesis on ATP generation or ATP synthesis in 1961. It is named as chemiosmotic hypothesis or chemiosmotic theory. Let us discuss about the ATP generation inside the chloroplast. This is a single thylakoid, and this is the membrane of thylakoid, and the thylakoid, it is the coin like structure present inside the chloroplast. Okay, and this is surrounded by a single membrane. It is not the double membrane, it is a single membrane. This is the inner surface and this is the outer surface, and this is the thickness of that membrane. Whenever we can see inside the membrane of thylakoid, there are two photosystems and electron carriers are present, as well as another part is present that is named as oxysome. Okay, and whenever the photon of light absorbed by these photosystems, that is photosystem 2 and photosystem 1, both are excited, and the reaction center of both the photosystem 1 releases its electron at the same time. Let us discuss from here, that is from the photosystem 2. Okay, when the photosystem 2, it can absorb the photon of light, the reaction center of photosystem 2 comes into the excitation state and release its electron, and this electron is first accepted by the primary electron acceptor, that is pheophytin, and this electron is high energy electron. Then this electron from pheophytin, it is accepted by another electron carrier, that is the plastoquinone. As the plastoquinone receive electron or accept electron, it is a proton carrier. Plastoquinone is a proton carrier. It is act as an proton pump inside the thylakoid membrane. As the plastoquinone receive electron from pheophytin of photosystem 2, it can accept proton from stroma of chloroplast and comes into a reduced state, that is PQS2 or PQH. Whenever the plastoquinone, it is a proton pump, or it is act as a proton pump, or it is a it is a proton carrier which is present in the thylakoid membrane. Whenever it can accept electron from pheophytin, at the same time it can accept proton from the stroma of chloroplast and get reduced into PQH. Whenever it can return back to its oxidized state, it can releases electron as well as proton. This electron is accepted by another electron carrier present in the thylakoid membrane, that is named as cytochrome B6F complex. As cytochrome B6F complex is an electron carrier, it cannot accept proton. So, it can push the proton into the lumen of thylakoid. This part, in inner part of the thylakoid, this is the inside of thylakoid, that is named as the lumen of thylakoid. And then the proton, it can pump into the lumen of thylakoid. And next, the cytochrome B6F complex. From cytochrome B6F complex, this electron is accepted by plastocyanin, and the plastocyanin is present on the inner surface of thylakoid membrane towards the lumen. And then from this plastocyanin, this electron is picked up by the reaction center of photosystem 1. As the photosystem 1 release its electron after comes into the excitation state. So this electron from the plastocyanin, it can fill the gap of the reaction center. And what about the reaction center of electron of photosystem 1? The electron which is released from the reaction center of photosystem 1, it can accept by the primary electron acceptor, that is the iron sulfur protein. Iron sulfur protein is the primary electron acceptor of photosystem 1. Then the electron from iron sulfur protein, it is accepted by ferredoxin, and ferredoxin is present towards on the outer surface of the thylakoid membrane towards the stroma. Okay, and from the ferredoxin, the electron is accepted by FNR, that is ferredoxin NADP reductase enzyme. Okay, and from the FNR, finally the electron is accepted by NADP, which is present in the stroma. Okay, and when NADP, it can receive electron, in the same time it can accept proton from stroma and get reduced into NADPH. Okay, what about the reaction center of photosystem 2? Now discuss about the reaction center of photosystem 2. It has also a deficiency of electron, and to fill the deficiency of reaction center of photosystem 2, a process takes place inside the lumen of thylakoid, that is photolysis of water. Photolysis of water takes place inside the lumen of thylakoid, through which the water molecule can be splitted into proton, electron and oxygen. Whereas the electrons can fill the gap or fill the deficiency of reaction center of photosystem 2. But what about the protons? As the protons are produced by the photolysis of water, it can accumulate inside the lumen of thylakoid. Okay? Due to the photolysis of water and due to the pumping of proton from stroma to the lumen of thylakoid by plastoquinone, these two regions can lead to the increase in the concentration of proton inside the lumen of thylakoid. And due to the use of proton from stroma, that is NADP can use the proton for its reduction, and again when the plastoquinone, it can pump the proton from the stroma into the lumen of thylakoid. These two regions leads to the decrease the concentration of proton in the stroma of chloroplast. Here the increase in the concentration of proton, here the decrease in the concentration of proton. Decrease the concentration of proton in the stroma and increase the concentration of proton in the lumen of thylakoid. This difference of proton across the membrane, it can develop proton gradient. And due to the increase in the excessive increase in the concentration of proton inside the lumen of thylakoid, it can generate a force that is named as proton motive force. And due to this force, the excessive amount of proton which is present in the lumen of thylakoid, they wants to move into their original place, that is the stroma. And these protons are moves from the lumen of thylakoid into the stroma of chloroplast through a special channel, through a special route is present in the thylakoid membrane, that is named as oxysome or ATP synthase, or it is also named as CF1CF0 particle. ATP सिंथेज भी बोलते हैं इसको, ऑक्सीजोम भी बोलते हैं. ओके, इट हैज टू सब यूनिट, one head part and one base part. This ATP synthase has two protein subunit. One protein subunit is embedded in the thylakoid membrane. It is named as CF1 particle, CF0 particle. And another part which is protrude towards the stroma of the chloroplast, it is named as CF1 particle. And whenever the high energy rich proton moves through CF0 particle of ATP synthase, the ATP synthase in CF1 particle get activated, and it leads to the conformational changes of this CF1 particle. And due to the conformational changes of CF1 particle resulting the binding of inorganic phosphate to ADP, एडिनोसिन डाइफॉस्फेट के साथ एडीपी आईपी क्या होता है, जुड़ जाता है, और क्या फॉर्मेशन होता है, एटीपी फॉर्मेशन होता है. And this can resulting the formation of ATP. Whenever the conformational changes occur in the F1 particle, it leads to the binding of inorganic phosphate to ADP and makes ATP. And इसी मैकेनिज्म के दौरान प्रोटॉन ग्रेडियंट बनता है, डेवलप होता है, तभी एटीपी का जनरेशन होता है. जब एटीपी ऐसे इस मैकेनिज्म के थ्रू जनरेट होती है, उसको बोलते हैं केमियोस्मोटिक हाइपोथेसिस और केमियोस्मोटिक थ्योरी, जिसको फर्स्ट स्टडीड किए थे पीटर मिटचेल ने. ओके. और इसको इस टाइप की जो जनरेशन होती है, जहाँ पे क्या होता है, हाइड्रोजन आयंस मूव्स फ्रॉम द रीजन ऑफ देयर हायर कंसंट्रेशन टू द रीजन ऑफ देयर लोअर कंसंट्रेशन अक्रॉस द थ्रू द सेमी परमिएबल मेम्ब्रेन और थ्रू द मेम्ब्रेन. इट इज नेम्ड एज केमियोस्मोसिस. यहाँ पे एक आयन का मूवमेंट हो रहा है, कहाँ से कहाँ पे, इन हायर कंसंट्रेशन टू लोअर कंसंट्रेशन में मूवमेंट हो रहा है थ्रू ए मेम्ब्रेन. सो इसको बोलते हैं केमियोस्मोसिस. और केमियोस्मोसिस के दौरान क्या होता है, एटीपी बनती है, एटीपी जनरेशन होता है. यहाँ पे एटीपी बन गया और यहाँ पे NADPH बन गया, जो कि एटीपी और NADPH दोनों का रिक्वायरमेंट होती है कहाँ पे, ग्लूकोज सिंथेसिस में. डार्क रिएक्शन जब होती है स्ट्रोमा में, ग्लूकोज सिंथेसिस होने की टाइम में, ये दोनों एनर्जी जो होते हैं, एनर्जी सोर्स ATP and NADPH2 are utilized during the synthesis of glucose. Okay, this is all about your chemiosmosis or chemiosmotic hypothesis. Thank you.